Hello, students. How are you? Good morning to everyone. And I am Rajveer Singh, your biology teacher. And today we are discussing brings the second part of the principle and inheritance. And I hope you will enjoy definitely previous session. And that was fantastic and conceptual for you. Okay, so let's start today's session. First question of the day, which is in front of you. In which of the following disorder, gynecomastia symptom is seen in individual? So first of all, we will talk about what is gynecomastia. So you know, gynec word is used just for female, okay, and mastia is the symptoms characteristics. So characteristics a disorder in which a gynecomastia symptom is seen in individual. So what? Down syndrome, Turner syndrome, Klinefelter syndrome, and phenylketonuria. So we know Down syndrome. We discussed already in yesterday's session about Down syndrome. So this is the trisomy. Okay. So this in the Down syndrome, gynecomastia is not present. Turner syndrome. Turner syndrome is present in female. So we will say gynecomastia symptom is not connected with Turner syndrome because basically gynecomastia definitely we can see and we will see in a male related disorder, right? So yes, Klinefelter syndrome, we have already discussed about Klinefelter. This is associated with male and we can say extra X chromosome, right? So gynecomastia symptom definitely connected with Klinefelter syndrome. Okay, phenylketonuria is another other type of disease. This is not related with gynecomastia, so that is why our answer is third. If I talk about conceptual approach behind of this answer, Klinefelter syndrome. So Klinefelter syndrome is a genetic condition that only affect male. As I told you, the condition is present from birth and is due to an extra X chromosome. Klinefelter syndrome is characterized by gynecomastia, a condition that causes boys and men's breasts to swell and become larger and then normal. Is it clear? In late puberty, okay, and hyalinization and fibrosis of the seminiferous tubules, elevated urinary gonadotrophin level, and the behavior concerns. Okay, so that was some symptom regarding of Klinefelter, and I hope you will understand. Klinefelter syndrome is related with male due to extra X chromosome, and what is characterized? So you can see at the screen, as I told you, right? This is very good question, right? Next question. Mark the correct option with respect to monosomy. Okay, so what it means monosomy, right? So as I told you, aneuploidy type is the monosomy nullisomy. Okay, as we discussed in yesterday's session, and those students who are seen first time my lectures, and you can see in previous lecture, part first of the principle and inheritance. So, what is the aspect monosomy regarding Klinefelter syndrome, Down syndrome, Turner syndrome, and hemophilia? Yes, the answer of this question very easy. So monosomy regarding correct option is the Turner syndrome. Okay, so answer will be third, right? So what is monosomy? Monosomy and nullisomy are types of the euploidy, right? And in other hand, if I talk about trisomy, it means is a 2n plus 1, right? So monosomy and nullisomy are type of a euploidy, and trisomy is a 2n plus 1. A chromosomal disorder in which a female is born with only one X chromosome, Turner syndrome, result from a missing or incomplete X chromosome. Symptoms include short stature, delay puberty. Characteristics are all regarding Turner syndrome, and infertility, heart defect, certain learning disability. Treatment involve hormones therapy in during Turner syndrome. Fertility treatment may be necessary for women who want to become a pregnant. Okay, I hope you will understand about the Turner syndrome's characteristic, the reason causing and the treatment therapy, right? Now, next, allosomic trisomy condition is seen in Turner syndrome, Klinefelter syndrome, Down syndrome, and this. So, as I told you, allosomic trisomy is not causing to Down syndrome. So, we can eliminate third and fourth option. Right now, we talk about first and second, Turner syndrome and Klinefelter. So, allosomic trisomy condition generally seen in the Klinefelter syndrome, not in Turner syndrome. Right. So, if I more talk about allosomic trisomy and Klinefelter syndrome, so in Klinefelter syndromes, there is a trisomy of allosome, that is, three sex chromosomes are present, and individuals suffering from Klinefelter syndromes has two X and one Y chromosome instead of the usual one X and one Y chromosome. Okay, so I hope you will understand. Now, next question: which of the following disorder is seen in human female only? Right? So what? Turner, Down, hemophilia, and Klinefelter. So as I told you, Down syndrome definitely can be seen male, female. Klinefelter definitely connected with male. Hemophilia, this is the autosomal disease, so can be seen male, female. Yes, but Turner syndrome is especially connected with female. So our answer will first, right? So, human female has 45 chromosome with XO. Turner syndrome is a condition in which a female is partially completely missing an X chromosome. So, a female with Turner syndromes has 45 chromosome, 45 plus XO. So that is why our answer is the first. Next question: when a pink flower Antirrhinum majus plant is test cross. Okay, do you know about test cross? Yes, test cross is a cross in which we cross a first one generation with the recessive homozygous parent genotype, right? So then phenotypic ratio in the resulting progeny is what? One red, one white, three red and one white, two pink and one white, one pink and one white. Yes, so we see, right, pink and white. This is a parental combination, and definitely they will see in 1:1, pink and white in a test cross, not red and white. Okay, so answer will be fourth. Okay, so now you can see at the screen. If I talk about in a table form, so when we cross, as I told you, F1 generation are crossed with the homozygous recessive genotype of a parent. So you can see small r and small r. And when we cross with F1 generation, then what we will be get? RR pink, small r white, R and small r pink, and RR white. Right? So Snapdragon Antirrhinum majus is a common name. Show incomplete dominance where both the genes express themselves equally in the heterozygous state. The genotype of pink flower RR, the genotype of white flower small r, and in above cross we will get 2 raised to 2, and when we simplify to this equation then we will get 1:1. Is it clear? So therefore pink and white flower are in equal proportions. So our answer will be fourth. Next question: heterozygous tall and violet flower pea plant were self and total 512 seeds are collected. That will be total number of seeds for both heterozygous trait is 288, 256, 384 and 64. So answer will be first 288. How can we get this figure 288? Because when we see the genotype of offspring produced after selfing of a heterozygous tall and violet plant is 9:3:3:1, where 9 represent the ratio of offspring with both heterozygous genotype of trait. Okay, and then after we will solve them. So then 9 we will get, and overall combination overall offspring is 16, and when we multiply with 5 and 12, then we will get 288. Seeds have both heterozygous trait. Okay, so that is why our answer is the 288. I hope you will understand. Next question. Mark the odd one with respect to F2 generation of a Mendelian dihybrid cross. Do you know about dihybrid cross? Yes, as we know, Mendel just did the hybridization cross. First monohybrid, second dihybrid, third is a trihybrid. Okay, so in monohybrid, Mendel just take a one character of a plant, pea plant. In dihybrid, he took the two characters, and in trihybrid he took the three character. Okay, so now then after what will our answer? Frequency of T and small t RR genotype is 12.5%. Frequency of TT and small r genotype is 6.25%. Frequency of both homozygous dominant genotype is 6.25%, and frequency of one homozygous recessive and heterozygous genotype is 25%. Yes, if I talk about odd one, so among of them odd one is the fourth, because if we see, then 25% we will not get genotype of the one homozygous and a homozygous recessive, and one is the heterozygous. Okay, remaining options are definitely right. So they are first, second, third. Why fourth is the odd one? Because in dihybrid cross is a between two different genes that have two different traits. As I told you, when described to dihybrid cross, right? When a tall plant with red color flower is crossed with dwarf plant with white colored flower, right? The F1 generation will have all tall plants with red colored flower, capital heterozygous, capital T small t, and capital R small r, right? When F1 generation is crossed with F2 generation, will have phenotype in the ratio of 9 raised to 3 raised to 3 raised to 1, where 9 parts will be tall plant with red flower, 3 will be tall plant and white colored flower, right? Then after colored, and 3 part will be dwarf plant with the red colored flower, and 1 part will be dwarf plant with white flower. The xenotypic ratio will be 1 raised to 2 raised to 1 raised to 2 raised to 4 raised to 2 raised to 1 raised to 2 raised to 1. In this frequency of capital T small t and capital R genotype will be, right? So this will be 12.5%, 12.5% frequency. 12.5% frequency of the TT and RR genotype will be 6.25% frequency of TT and RR genotype will be 6.25% frequency of TT and RR genotype will be 12.5% frequency of this, right? Will be. Okay, do you know how can we get this frequency? When we see in the previous question at which we talked about, when we calculate overall offspring and a particular offspring we will get in this combination and multiply with overall plants, right? Overall bird. Then after we can get the frequency of the following, just like 12.5%, 6.25%, right? So now frequency of this will be 12.5%, frequency of TT and RR will be 12.5%, frequency of TT and small r will be 6.25%, and then after frequency of a TT and small r will be 25%. Frequency of this will be this. So as I told you, among of them, now you will understand why we have choose the odd one option. Fourth is the odd small t and capital R small r genotype reference. Okay. Now next question. Next question after that, find out the frequency of A, B, B, C and small c, D and small d, E, E. If parents are, right, if parents are this and this, so what will we get, right? So as I told you, first of all we will find out the heterozygous form, and then after we will solve this question. Okay, so answer this question will be second, but how this value will come? So according to the law of independent inheritance, the allele are inherited independently. Okay, hence, the given questions, each gene is analyzed separately. Okay, as I told you, first of all. So when we cross with capital capital A, small a in heterozygous form, then 1 by 4 of capital A, and 1 by 2 in heterozygous, and homozygous recessive is 1 by 4. Hence, what we see, half, it means 50% offspring will have capital and small a heterozygous form. Then after, similarly, small b and small b, all are small b. It means all spring will have small b. Small c and small capital C, all will definitely get in heterozygous form. That means all spring will have capital C and small c heterozygous form. Then after we will talk heterozygous form and dominant, recessive homozygous phenotype, so genotype, and when we will cross among of them, then what we get, half of heterozygous and half of homozygous recessive. So what it means, half, 50% offspring will have a heterozygous form. Then after, when we cross fifth number in heterozygous and homozygous recessive. So half, 50% will be half of E and E, and half of E into E. Hence, half or 50% offspring will have EE, right? So hence frequency of this will be half into 1, 1 into half. So when we multiply with all data, then we will find 1 by 8, and 1 by 8 it means which is equal to 12.5%. So hence in this manner we will get our answer 12.5% in second options. Next, incomplete dominance. What it means, dominant trait is completely expressed in F1 generation. Phenotype and genotype ratio are different. Two dominant alleles are need to express the complete dominant trait. F1 individual have the equal traits for both parent. So what we see, among of them, if I define incomplete dominance, so third option is the suitable for definition to incomplete dominancy, because two dominant allele are needed to express the complete dominant trait. Okay, so that is why our answer will be third, right? So here, if more talk, when none of the factors of a gene is dominant, the phenotype of heterozygous dominant individual is a blended of a dominant and recessive trait. This is called incomplete dominancy. For example, flower color in Mirabilis jalapa. The red flower is the dominant character and the white flower is the recessive character. Right? This is, that is blending of a two allele. The flower in the F1 generation will be pink in color due to the mixing of the allele. Right? Next question, progeny with blood group O cannot be obtained in cross in which following? A and A and B, O and AB, and B and B. Definitely our answer will third. Why? Because as I told you in same type question yesterday, at where I talked to you, antigen blood group O, it means it's a show just a denote 0, no any antigen, right? And where, and if any no any antigen present in blood group O, then definitely other any type blood groups can show their effect. Okay, whatever blood group A, B and AB. Right? So that is why our answer will third. Now you can understand also a child of blood group has a parent with blood group A and A. B, B, A and O, B and O, but not A and B, B and AB. So the correct answer is the, or we have choose the third option. Okay, because in that particular combination, blood group will not be O in any case. Right. Now next question: which of the following parental combination has a produced mutant offspring? T, small t, then we'll get heterozygous, homozygous and homozygous, then heterozygous, homozygous, heterozygous, and sorry, heterozygous, homozygous, and heterozygous, sorry, homozygous, dominant and recessive, then heterozygous. So when we can see clearly in second option, small t, it means a homozygous recessive. But what we are seeing result? In result, we can see, we are just seeing to heterozygous form and in which small capital T are present, but in parental combinations, not any capital T present at anywhere. It means this is the mutant offspring. So that is why we have choose the first, second option. Sorry, right? Okay. So first of all, you will understand about the what is the concept to choose this option, right? But here I would like to say about sexual reproductions, variation and mutation. So sexual reproduction is the reproductions which is caused by the fusion of gametes. The male and female gametes fuse to form the zygote, right? Due to the fusion of gametes, the product of sexual reproduction generally generate new genetic combination leading to a variation. The organism develop from the zygote which has characters of both the parent. The genome of the offspring is formed by the recombination of genetic material. So they are also known as a recombinant. The recombination may cause the sudden heritable changes in the gene pool which cause mutation. So our answer is the second. Right? Next, which of the following combination seems to have linkage in character selected by Mendels? Stem height and pod color, flower color and flower position, seed shape and seed color, plant height and pod shape. So basically, linkage is can observe by in Mendel character in a plant height and pod shape because the gene of this character are present at a same chromosome. So that is why plant height and pod shape these are character. So this can show a linkage. Okay. So the chromosome number 4 of pea plant carries a gene for flower position, pod form and plant height. Out of this, out of these three genes, the two gene, namely the gene for pod form and one for the plant height, are closely placed in on the chromosome that they show a linkage. However, the gene for flower position is located far apart from the other two gene. So do not show linkage, but this will show the linkage. Okay. So I hope you will understand why we have choose this option. Okay. So now we come at next question. Next question is a diploid organism. A diploid organism is heterozygous for 5 loci and homozygous. are diploid organisms in heterozygous for 5 loci and homozygous for 2 loci. How many types of gametes can be produced? 1 to 8, 32, 4, and 14. So definitely, this is a very simple question. So you can get answer. But what is the concept behind of this answer? So we will discuss. So answer will be second, 32, right? So types of gametes can be calculated by the formula of the 2n, where n is the number of the heterozygous loci. In the given case, in the given case, there are 5 loci heterozygous, as I told you, and 2 loci are homozygous among of them, right? So now, when we solve, so first of all, we will choose the formula 2's power n, and here we will put the value n is the number of the heterozygous, right? So here, and as we know, here heterozygous form is the number 5. So at the place of the n, we will put the value 5. And when we solve this question, so 2's power 5 is the 32. Okay, so our answer will be 32, right? Now, next question, a Lesch-Nyhan disease in an X-linked recessive disorder that cause neurological damage, right, in human beings. A survey of 500 mets from a Caucasian population revealed that 220 were affected with this disorder. What is the frequency of the normal allele in this population? So our answer will be second, point 0.8, 0.8, right? Why? Why? Because total individual 500, affected individual among of them 20, right? Then after, Lesch-Nyhan syndrome is a X-linked recessive disorder, which means that the gene mutation is carried out by the mother and pass on to her son. Female are the carrier in this disorder. Now, according to Hardy-Weinberg's principle, p square plus 2pq plus q square is equals to 1, and where p is the frequency of dominant allele, q is the frequency of the recessive allele, and p square is the percentage of homozygous dominant allele, right? Q square again is the percentage of the homozygous recessive allele. 2pq is the percentage of the heterozygous allele. So a single p just show to frequency, and p square will show to a percentage. Here, in this case, frequency of the recessive allele p is q square, sorry, 20 upon 500, and when we solve, then we will get the value 0.05, 0.04. So that the value of q is 0.28, 0.2, 0.2, right? And frequency of the dominant normal allele p, right? So as we know, p plus q is equals to 1, and when we solve the value of p, so we will get the 0.08. Okay, so our answer will be second, 0.08, 0.8. I hope you will understand. Next question, how many type of the zygotic combination are possible between a cross A and small a, BB, capital C and small c, DD, right? And between with the capital A, small b, capital C and small c, capital D and capital D. So what will we get the answer? Answer will be 128. But how the answer will be get 128? So first of all, number of gametes possible in the parent is a 2n, where n number of the heterozygous allele in a parent genotype. Number of zygote possible are the gametes into gamete. The number of gamete formation possible by the parent one, right? As this gender is shown in question part. Then here we can see Heterozygous part is third, right? So which I write down in the bracket. Okay. So then after when we solve 2 power 3, and so when we will get 8, answer will be 8. N is the number of a heterozygous allele, and is the oral value which is shown in the 3. The number of gamete formation possible by parent to AA, BB, CC, and DD, and 2 power 1. So here answer will be 2. N number of heterozygous allele is a 1. So the number of zygote possible are gamete of parent 1 and gamete of the parent 2. And when we solve, so 8 into 2, we will get our value that 16. Okay. Now right, so our answer here in next question, in F2 generation of a Mendelian dihybrid cross TTRR, small r, tall plant and violet flower plant are obtained in 1:1 frequency. Ratio of parental and non-parental plants is 1:15. Recombinant plant are obtained 1:1 frequency. So among of them, more than one option is correct. Yes, our answer will be 4. Option more than one is the right. Okay, so first of all, we will talk about dihybrid cross of the Mendelian in F2 generation. So in the given question, tall plants T are dominant over shoot plant small t. Similarly, violet flower small r are dominant over the white flower small r. The gametes produced by small t, sorry, capital T, capital R, and small t, small r. Plant will be T and R, and here in other hand, small t and small r, respectively. The genotype of offspring of F1 generation will be capital T, capital R, and small r, and the phenotype will be tall and violet flower plant. When this F1 generation, F1 is crossed, the F2 generation will have phenotypic ratio of 9 raised to 3 ratio 3 raised to 1, where 9 part is the tall and violet flower plants, and 3 parts are tall and white flower plants. 3 are short and violet flower plant, and 1 part is a short and white flower plant. Hence, in F2 generation, 12 parts will be tall and 12 parts will be violet flower plant. That is, their frequency will be 1:1. It means test cross. The ratio of parental plant, tall and violet flower is short and white flower, and non-parental plants, tall and white flower plant, and short and violet flower will be 10:6. So that is why we have choose the fourth option. Okay. Next question is a Morgan hybridized yellow bodies, white eyed female to brown bodies, red eyed male, and intercrossed their F1 progeny. He observed that what? F2 ratio was deviated very significantly from the 9:3:3:1 ratio. Both genes did not segregate independently for each other. Recombinant type are not obtained in F2 generation. Both genes segregate independently of each other. So, what is the correct side of statement? Yes, among of them, statement A is definitely right. We saw in previous question both, and second, right, and then after both genes did not segregate independently for each other. This is also right, but recombinant type are not obtained. This is absolutely wrong because these are present, we can say obtained in F2 generation, and both genes segregate independently of each other. This is also wrong. So what our answer is the first A and B only correct set of the statement, right? So when the gene of certain feature like yellow white body or white red eye Drosophila are located very closely to each other on a chromosome. There are a little chance of a crossing over, giving higher parental combination. When the genes are on different loci on a chromosome, the distance between them considerable. There are higher chance of a recombination, giving rise to mixed or non-parental features for autosomes. However, since body color and eye color of parents are present on X chromosome, the deviation will be relation to gender in addition to recombination, which will not occur in male as they in homozygous for the traits they carry. Okay, so that is why our answer is the first statement is being right. Next, A dash used the frequency of recombination between gene pair of dash B as a measure of the distance between genes and mapped their position on the chromosome. Right. So, as we know about Struven, this who was the student of the Morgan, right? Proposed the chromosomal map. Okay, so so our answer will be first. Student A is student use the frequency of the recombination between gene pairs of the same chromosome. Okay, as a measure of the distance between the gene and map their position on the chromosome. So that is why our answer will not second. Answer will be fourth, because in option second student also present, but you can say different chromosome are present. Here write down, but here chromosome was the same, not a different. Next question, correct option is the student, as I told you, explain you, so you can see also. Okay, now next question. While solving the problem of the sex determination in a large number of insect, it is observed that all egg lacks sex chromosome. Some of the sperm bear the X chromosome. All eggs as well as sperm bear the X chromosome. Some of the egg bear the X chromosome. So answer will second. Some of the sperm bear the X chromosome. Why we have choose only second option? Because this structure is a sex chromosome, or usually known as a X chromosome in an insect. A lot of insects have XO type of the sex determination, where female have XX while male have single X chromosome, denoted by X and 0, and we can say in other words XO. Hence, after spermatogenesis, only 50% of the gametes have this structure X chromosome, while other 50% are without X chromosome. Sperm with X chromosome fuse with ovum, resulting in female offspring, while those without X chromosome fuse with ovum to form a male offspring. So that is why our answer is second. Right? Now, next question. Loss or gain of a segment of DNA result in frame shift mutation, point mutation, polyploidy, chromosomal aberration. Definitely, chromosomal aberration is the right answer regarding of the loss or gain of the segment of DNA result in, because polyploidy Polyploidy is the state of a cell or organism having more than two paired homologous sets of a chromosome. A deletion is a mutation in which a part of a chromosome or a sequence of DNA is a loss during DNA replication. Chromosome inversions is a condition in which a chromosome segment is clipped out, turned upside down, and reinstated back into the chromosome. Transversions are interchanges of the purine for pyrimidine base, right? Purine replaced by pyrimidine in transversion. In transition, purine replaced by purine and pyrimidine replaced by pyrimidine, right? during transitions, and transmission is just a reverse, which therefore involve an exchange of one ring and structure. Duplication is a particular kind of the mutation involving the production of a one or more copies of any species, any piece of DNA, including sometime a gene or even an entire chromosome. So that is why our answer is the fourth. Okay, now next question: which one of the following is a physical factor that induce mutation? Acridine, nitrous acid, HNO2, UV light, UV rays, and base analogs. Yes, so we can see acridine, HNO3, and base analog. They are definitely mutagen, but they are chemical. But if we talk about physically, so physical is the UV radiation, UV rays, ultraviolet light, and ultraviolet radio rays. The most common environment factor that cause mutation is ultraviolet rays. They penetrate deep into the cells of an organism and make alteration in or mutate the genetic material. So our answer is the third. Next, the chromosome map are not accurate map because crossing over frequency is higher than recombination frequency. Yes, this is right. One crossing over, there, one crossing over interface and increase the frequency of increase frequency of nearby crossing over frequency decrease towards the end of the chromosome. Heterochromatin increase the means crossing over. So our answer will be first. Crossing over frequency is higher than recombinant frequency. So, if I more talk about the behind of this question, behind of this concept, so the chromosome map are not accurate map because, as I told you in question part, one crossing over interface and increase the frequency of the nearby crossing over. The recombination at one place increase or decrease the chance of the recombination in the nearby place. This is because crossing over at one place causes steric hindrance. This interface with the genetic mapping. Okay, so that is why our answer is the first. Next question: if a agouti mice CCAA is crossed with albino mice CCAA, then how many albino mice are produced in resulting progeny? Right, so 4, 9, 2, 3. Answer will 2. How? So you can see the coat color of mice is an example of recessive epistasis. Recessive epistasis occur when the recessive allele of the one gene dominate the allele of the second gene. In mice, the wild type coat color, it means agouti is dominated over the albino coat color. When there is a cross between the agouti and albino mice, it means the cross of progenies CCAA and small c and capital A, capital A. It give it gives the result of progeny with this combination. In this cross, the phenotypic ratio would be this, and here 9 belong to the agouti color, 3 belong to solid color, and 5 have the albino color. Therefore, a cross between CCAA and small c and capital A. Right, so would generate four type of offspring, which is the genotype this and this. Okay, here two are agouti and the other two are albino. The mice with the genotype CC will have the albino coat color. So here in this question, there will be two progeny produced or 50-50% progenies. So parent will this, gamete will this, right? So now if we see in table format F1 generation, so we can see four type of gametes, and in vertical column downside CA and CA, and this will the combination. Okay, so our answer will be second. Two type, number of two will be answer. Next, epistasis and dominance are respectively intergenic, non-allelic, extra-allelic, non-allelic, inter-allelic, intergenic, non-allelic. So answer will be third. Non-allelic and inter-allelic. Okay, right. So why we have choose third option? Because in epistasis, one gene pair masks the expression of the another pair of gene. Epistasis gene is a gene that determine whether or not a trait will be expressed. The system of gene that determine skin color in a man, for example, is independent of the gene responsible for albinism, lack of pigments, or the development of skin color. This gene is an epistatic gene. When the albino condition occur the gene that determine the skin color are present but not expressed. And if I talk about dominance, so dominance is the greater influence by one of the pair of a gene allele that affect the same inherited characters. If an individual's pea plant with the allele capital T and small t, capital T tallness and small t shortness is of the same height as a T and T individuals, right? So the is capital T alleles and the trait's tallness is said to be completely dominant if the capital T small t individual is shorter than the capital T and capital T, but still taller than the small t individual. Capital T is said to be partially or incompletely dominant. It means it has a greater influence than small t but does not completely mask the presence of small t, which is said to be recessive. Thus, the correct answer is the option third. Right. Next question. In a next question is in a complementary gene, interactions calculate the number of the phenotypes and genotype produced in cross A in cross AA and BB and small capital A, small a, capital B, capital B. So one phenotype, two phenotype and four genotype, four phenotype and four genotype, two phenotype and two genotype. So answer will be two phenotype and four genotype. Why? Because in complementary gene interactions, when both dominant allele are present together, they produce a new phenotypes. In this type, AA, capital B small b, and capital A, capital small b, and capital A, capital small b, genotype produce similar type of phenotype. The genotype this and this and this produce one type of the phenotype, right? Hence, when a cross is made between capital A, capital B, small a, capital B, the gametes produced will be capital B, capital A, capital B, small b, capital and small b, capital small a and capital B, small a and capital B. In this offspring, will have genotype heterozygous and that's dominant, homozygous, heterozygous and heterozygous, and homozygous recessive, homozygous dominant, and two type of phenotypes will be observed. Okay, so hence you can see now, and I hope you will understand two type of phenotype and four type genotype we will get by this cross, by this following cross phenotype and genotype. Okay. Next question: how many type of gametes will be produced by male Drosophila having following arrangement of two gene, this and this on X chromosome? Okay. So what? Answer will second two. Okay. Because the genotype given consists of one X chromosome with two gene and one Y chromosome, During gametogenesis, these two chromosomes will segregate. Okay, during gametogenesis, these two chromosomes will segregate. Hence, the gamete produced formed will be one X chromosome and autosome, and second with the Y chromosome and autosome. Okay, so that is why our answer is the first. Next, if interference is a complete or set percent, then the frequency of observed double crossover will be equal to expected, greater than expected, lesser than expected, 0. Okay, so answer will be 0. Now you shocked. How can it possible? 0. Okay, so 0 is answered because in interference the complete or 100%, then the frequency of the observed double crossing over will be 0. This is because a crossover in one region will completely block the crossover in adjacent region. In some region of the chromosome, there is never any observed double crossover. Here, the coefficient of coincidence is a 0, and interference is a complete. Also, interference I 1 minus observed double crossover. If interference is a complete, then the observed double crossover will be 0. So that is why our answer is the fourth. Next question. In the given pedigree, indicate whether the shaded symbol indicate dominant or recessive. So as I told you, and you see also, you saw also in theoretical part of pedigree analysis, if the characters are present in every generation, then definitely this type character will dominant, not recessive. And when a generation is skipped, then definitely this character will mention as a recessive. So you can see in diagram, every generation is infected by following disease. So we can say this is a dominant disease. Answer will be third. Okay. In the pedigree chart, the shaded figure refer to the dominant allele, as I told you in question part. And because irrespective of the sex of the offspring, that is trait expressed, which is only possible, it is dominant in nature. Okay. So our answer will be third. Next, in which of the following disorder a single protein that is a part of the cascade protein involved in blood clotting is affected? Thalassemia, sickle cell, hemophilia, phenylketonuria. Answer will hemophilia. Why? Why we choose hemophilia, not to thalassemia, sickle cell, phenylketonuria, PKU? Because hemophilia is sex-linked disease. Okay, and a single protein that is a part of the cascade of the protein is involved in the clotting of the blood is affected. The heterozygous female for hemophilia may transmit the disease to sons. Okay, so now next question, mark the correct statement with respect to sickle cell anemia. Okay, so what homozygous individual for HB hemoglobin S are apparently unaffected. This is wrong because when individual are homozygous, then definitely is affected. So we can say this statement is incorrect. Next, heterozygous individual exhibits sickle cell trait. Yes, this we can say this statement is right. Next one, heterozygous individual are affected as well as carrier. No, it's absolutely wrong because when individual are carrier, then it is not affected. It will be unaffected. So hence our statement third is also wrong. Homozygous individual for him HbA show the disappeared phenotype, disease phenotype. Not right. Because homozygous individual of HbA does not show the disease phenotype. So what we see? Option fourth is also wrong. So our answer will be second. Okay, so you can see here heterozygous individual exhibits sickle cell trait is the right statement. Remaining are wrong. Why is wrong? So as I told you in a bracket part, this will affected, not unaffected, and third part will unaffected, not affected. So this also wrong. And fourth part you can see not show. In this part question they are saying it will show, but this is wrong. It does not show. Okay, so that is why our answer is second. Okay, so dear students, that was today's sessions. Thank you for watching to me, and really I am very happy to read and teach this topic by fantastic and some good questions. Okay, so thank you Bye bye. Let's we meet tomorrow. New topic. So topic will sell new questions, new session. Okay. Bye bye. Keep a smile.